Hello, my name is Zosia Maciorowski. I'm with the Live Education Delivery Subcommittee of the ISAC Education Committee, and today I'm going to talk about the basics of flow cytometry. In this talk, we'll discuss what flow cytometry is, what parameters we can measure, light scatter and fluorescence, and then we'll walk through the inside of a flow cytometer and see how it works. The word cytometry comes from the Greek. Cyto means cell, metry means to measure. So cytometry measures cells or other particles such as bacteria, algae, vesicles, or beads. Let's start with the light microscope, which I think you're all familiar with. In a light microscope, the light source is a lamp. Our cells, or whatever, are on a microscope slide, and we look at them by eye through the microscope's ocular. In a fluorescent microscope, the light source can be either a laser or a lamp, illuminating the microscope slide with our cells, which are stained with a fluorescent marker. We can either look at the cells by eye, via the microscope oculars, or with a camera, which has the advantage of recording the images for further analysis and quantitation by dedicated softwares. Flow cytometry is cytometry where the cells are measured within a flow of liquid. It's a technology that allows us to measure light scatter, fluorescence intensity, on cells or other particles, and one by one, the cells or particles must be in suspension. Here in a nutshell is how it works. We have a flow cytometer. The light source is a laser, which intersects the cells as they pass one by one in a flow of liquid. The cells are stained with fluorochromes, which then fluoresce, and the emitted light is captured by a light detector, which produces a voltage pulse proportional to the amount of emitted light. The voltage pulse is digitized to a channel number and saved in a data file, and some of the newer instruments also give us an image of the cell. The advantages of flow cytometry are that it's very high speed. You have data on millions to tens of millions to sometimes hundreds of millions of cells, can measure many parameters simultaneously, which gives high statistical significance and allows you to detect rare populations. If you have a cell sorter, you can sort out the cells of interest so you have pure populations for downstream experiments. Classically, flow cytometry has been used to measure particles from about 1 to 30 microns. Some instruments allow the measurement of larger elements, up to 1.5 millimeters, clusters of cells like organoids and spheroids, organisms such as C. elegans or zebrafish, seeds, nematodes, insect eggs, and larvae. And we can now measure very small particles as well, down to 50 nanometers, small bacteria and viruses, and particularly of interest now with all the work being done on extracellular vesicles. Now we'll talk about what we measure by flow cytometry, light scatter and fluorescence. Here we have a mix of unstained cells of different sizes, textures, and granularity. There are light scatter characteristics on this graph, forward scatter on the x-axis and side scatter on the y-axis, allow us to identify different cell populations based on their size and texture, and we'll look at this in detail in a minute. Fluorescence, here we have a mix of cells, again different sizes, now also stained with red and green fluorochromes attached to different markers. We can look at the different populations identified by their scatter. Here we're looking at the fluorescent parameters of the lymphocytes only, and then delve into the expression of those markers on the different subpopulations of lymphocytes. The wavelengths of light used in flow cytometry range from the UV, down around 350 nanometers, up through the violet, blue, green, yellow, and red, up to the far reds at around 800 nanometers. You may remember this from your basic physics classes, but keep in mind that the lower or shorter wavelengths have higher energy, and the higher or longer wavelengths have less energy. Let's go into a little more detail on the light scatter. A cell or particle passing through a laser beam refracts or scatters light in all directions. In flow cytometry, we measure the scattered light at two angles. The forward scatter, or FSC, is measured in the axis of the laser beam. Notice that there's a bar in front of the light detector. This is called an obscuration bar, and it blocks the laser beam itself, so that the light that is measured is only the light scattered in that direction by the cell or particle. The side scatter, or SSC, is measured at a 90-degree angle to the laser beam. And what does light scatter tell us? The forward scatter gives an indication of the size and the surface properties, while the side scatter is dependent on the structural complexity and granularity of the cells or particles. So here we have a typical scatter plot with the forward scatter measurement on the x-axis and the side scatter on the y-axis. This is a peripheral blood preparation with three populations that are different on their forward and side scatter parameters. We have lymphocytes, monocytes, and granulocytes, and we can separate them out quite nicely because the little clouds of cells are quite distinct. Each dot represents a cell. It's a very useful parameter to distinguish different types of cells based on refractive index, size, surface properties, and internal complexity. And we use scatter in every cytometry experiment, but neither forward scatter nor side scatter can be used to quantitate exact size. Now let's delve a little more into fluorescence and fluorochromes. Fluorochromes are molecules which absorb light at one wavelength and then re-emit the light energy at a longer wavelength. Generally, they're low molecular weight, flat, multi-ringed structures. Here we have the structure of one of the most commonly used fluorochromes, fluorescine or FITC. You can see its series of rings. And here we have phycoerythrin or PE. It's the phycobilin chromophore part that absorbs and emits light. Multiple phycobilins are bound to proteins to make quite a large molecule. Here is a simple schema showing how fluorescence works. We have a fluorochrome molecule with an electron at a stable resting level. The electron is excited, absorbing the energy from a blue 488 nanometer laser, and jumps to a higher unstable energy level. It then loses some of the energy non-radiatively through heat, and the rest of the energy is emitted as fluorescent light at a higher wavelength with lower energy. On this rainbow scale of light wavelengths, we can see that the excitation wavelength at 488 is shorter but higher energy than the longer emission wavelength with lower energy. The difference in energy between excitation and emission wavelengths is called the Stokes shift. This is very simplified. It's the fluorochrome's electron cloud that absorbs and emits energy. Each fluorochrome is capable of absorbing light energy across a specific range of wavelengths. That's its excitation spectrum. Here we're looking at the FITC excitation spectrum. FITC can absorb energy at all of these wavelengths, but it absorbs best at its excitation max, which is around 495 nanometers. Similarly, each fluorochrome emits light energy over a specific range of wavelengths. That's its emission spectrum. FITC will emit fluorescence at all of these wavelengths, but most efficiently at 520. If we overlay the excitation and emission spectra, we can see that the emission spectrum is always at a longer or higher wavelength with lower energy than the excitation spectrum, and it typically tails off to the right into the higher wavelengths. Laser light is used to excite fluorochromes on a flow cytometer, and these are the lasers found on standard flow cytometers. Originally, cytometers had a 488 nanometer blue laser and a 633 nanometer red laser. More recently, a 405 violet laser is becoming standard on a 3-laser instrument. The high-end 5-laser and up instruments now have added 561 yellow-green laser and a UV or a near UV laser that can be 355 to 375 nanometers. Fluorochromes can be excited by multiple lasers. Here the dotted lines are the excitation spectra and the filled are the emission spectra. Here we have FITC, which is optimally excited by the 488 laser, but you can see that it's also excited to a much lower extent by the 405 and the UV laser. This is called cross-laser excitation. Allophycocyanin, or APC, another commonly used fluorochrome, is maximally excited by the 640 red laser and to a lesser extent by the 561, 488, 405, and 355 UV lasers. We will see the extent and importance of this cross-laser excitation a little later when we talk about spectral cytometry. Here are the emission spectra of 3 fluorochromes, FITC, PE, and PE Texas Red, all excited by the same 488 laser. Notice that the peaks of the individual emission spectra are distinct and pretty well separated, although they do tail off to the right, so there is some overlap. Here, however, we have added in a fourth fluorochrome, PECF594, which has an emission spectrum that almost completely overlaps the PE Texas Red spectrum. The light these two fluorochromes will emit are at the same wavelengths, so cannot be differentiated on a conventional flow cytometer. The take-home lesson here is that fluorochromes are often excited by multiple lasers, and their emission spectra can also significantly overlap. Choosing fluorochromes carefully to avoid the spectral overlap in order to allow the accurate measurement of each fluorochrome is essential. How do we use fluorochromes in flow cytometry? One of the most common ways is phenotyping cells with fluorochrome labeled antibodies, where we want to know how much or how little of a certain antigen or marker is expressed in or on our cells. Here's our cell with a surface antigen that we want to measure. We have an antibody that binds specifically to that antigen, and the antibody is coupled to a fluorochrome so that the amount of light emitted by the fluorochrome as the cell goes through the laser gives us information about how much antigen is present on our cell. Looking at a graph of the number of cells on the y-axis versus intensity of fluorescence on the x-axis shows us that we have two populations of cells here, one with low fluorescence on the left, cells with few or no antigens, and a large population on the right, cells with high antigen density. Here again we see a cell labeled with antibodies to two different markers. Antibody A is coupled to a green emitting fluorochrome, and antibody B to a red one, so we can quantify these markers separately. Fluorochromes called tandem dyes are commonly used in cytometry. Tandem dyes are two fluorochromes that are tightly coupled together. This example is a PE-CY5 tandem. PE is the donor or base fluorochrome, and CY5 is the acceptor. The donor fluorochrome is excited by the laser, but instead of emitting the energy as fluorescence directly across its emission spectrum, it transfers that energy non-radiatively to the acceptor fluorochrome. The acceptor's excitation spectrum must significantly overlap the donor's emission spectrum. The acceptor fluorochrome then re-emits that energy at its own higher emission spectrum. Here we see the excitation and emission spectra of PE, emission max around 570, and CY5, emission max around 670. If we overlay these, we can see that the emission of PE significantly overlaps the excitation of CY5, so that PE can transfer that energy to excite the Cy5 molecule, which then emits at its own emission wavelength. Depending on how tightly the fluorochromes are coupled, there is often incomplete energy transfer with some leakage of fluorescence from the donor, here PE, at its emission wavelengths. Another commonly used application of cytometry is the identification of gene transfected cells using fluorescent proteins. Transfection of genes of interest into cells in order to examine the effects of that gene are not always highly successful. Sometimes as few as 1% of the cells are successfully transfected. However, if the gene that codes for the protein of interest also contains a reporter gene which produces a fluorescent protein, here it is the green fluorescent protein, GFP, the cells that have successfully taken up the gene of interest will fluoresce green. The number of successfully transfected cells can be quantitated and, more importantly, sorted using a flow cytometry cell sorter to obtain a pure population of cells expressing that gene for downstream analysis. Many fluorescent proteins are now available in a rainbow of colors. In addition to antibody-coupled fluorochromes and fluorescent proteins, There's a huge range of fluorescent probes available for cellular components and processes on the cell surface and inside the cell cytoplasm and nucleus. We can measure DNA content and cell cycle phases with DNA binding dyes, cell proliferation. There are probes that are pH sensitive, measure calcium flux, membrane potential. Some are organelle specific or identify dead cells. Others measure oxidative states, phosphorylation, and numerous activation markers, and this is just to name a few. Some cells, such as algae, contain natural fluorescent pigments that can be used to identify different species. A vast array of applications, both research and clinical, use flow cytometry. On the research side, cell biology and immunophenotyping are among the most common. Also of importance are the applications in marine biology, plant biology, particularly for DNA ploidy, microbiology, and extracellular vesicles. Clinically, flow cytometry has been central in the diagnosis and prognosis of leukemias and lymphomas, as well as assessment of MRD, or minimal residual disease, but it also encompasses a wide range of other clinical applications, a few of which are shown here. Now let's take a walk through the inside of a flow cytometer and see how it works. It's not a black box. It's important to understand how an instrument functions so that you can generate good, reliable, and reproducible data. The quality of your data will be dependent on how well you know your instrument and how well you set up your experiments to make the best use of it. Here we can see the inside of a BD fax caliber. It's a simple 2-laser 4-color cytometer that we'll use as a model in our talk. The caliber is one of the early user-friendly BD workhorses and is still in use in many parts of the world. The newer instruments may have more lasers and colors and fancier electronics, as we will see, but the main principles are pretty much the same. Inside a flow cytometer, we have the fluidics that position cells to flow one by one past the laser beam. The optics separate the light emission from the different fluorochromes and direct them towards detectors, and the electronics, which convert photons of light emission to voltage pulses, which are amplified and digitized, and then stored in the computer as a data file. Let's look first at the fluidics. This is a very basic fluidic schema of a typical positive air pressure system. We have a sheath tank and a waste tank. Here are tanks from different cytometers. At the bottom are the caliber tanks. In the tanks, we have sheath fluid, which can be a saline solution, PBS, water, or it can be a buffer adapted to your cell type. At the bottom in the center here, we have the sample tube with our cells in it. Above it is a conical sheath chamber, which directs the sheath and the sample towards the flow cell, where the cells pass one by one through the laser beam. Let's look at how it works. The sheath tank is connected to the sheath chamber, and the waste tank connected to the waste outlet. An air pressure pump pressurizes the sheath tank so that the sheath fluid is forced out of the sheath tank and into the sheath chamber and up through the flow cell. The sheath pressure must be very stable, and it's tightly regulated by the instrument. Another pump pressurizes the sample tube. The sample pressure is slightly higher than the sheath pressure, and it forces the cells from the sample tube up into the sheath fluid in the sheath chamber, forming a central core within the sheath fluid so that the cells pass one by one through the center of the laser in the flow cell. This sample differential is controlled by the user, and we'll talk more about that in a few minutes. This is a very simplified schema. Cytometers have complex regulation mechanisms to control the sheath and sample delivery. On the left, we have our schema again. In the center, we can see it in more detail. The sheath fluid input, the sample air pressure forcing the sample into the sheath chamber. And on the right, we see the real thing. Again, the sample tubes at the bottom, the conical sheath chamber, and the flow cell, and our laser is on. There are a number of ways to push the sheath fluid and the sample through the cytometer. We've just looked at positive air pressure used by many of the BD instruments, the Caliber, of course, Fortessa, the Symphonies, the ZE5 sheath fluid, and most sorters, Astrios, the Arias, Discover S8, and the Aurora sorter. A syringe pump is used in the Guava for the sample in the Attune and in the Novasight series of instruments. Peristaltic pumps are common as well, in the Cytoflex, the Novasight sheath, ZE5 sample, and the Accuray, for example. And finally, they can be vacuum-driven, as in the Aurora analyzer and the BD Fax Lyric. A crucial element of the fluidics is to center the cells in the stream as they pass through the laser. This is done using hydrodynamic focusing, so that there is a central stream of cells within the stream of sheath fluid. The shape of the sheath chamber and flow cell, and the differential pressure between the sample and the sheath fluids create a uniform profile of parallel layers called laminar flow. The sample pressure is slightly higher than the sheath pressure. The sample stream is injected into the sheath stream, and the sheath fluid exerts pressure on the incoming sample stream so that the cells are forced into a central core and pass one by one through the center of the laser beam. Certain instruments, like the Attune, use acoustic focusing in addition to the hydrodynamic focusing, which allows the cells to be well centered at a high throughput. Here we can see fluorescent yellow beads passing through the flow cell. The laser is on. The sheath pressure is off, so there is no hydrodynamic focusing, and we can see that the beads are spread all across the flow cell as they pass through the laser beam. Here the sheath pressure is on, and we have hydrodynamic focusing, forcing the beads into a nice narrow core in the center of the stream and of the laser beam. Where the laser intercepts the cell in the stream differs between instruments. On the left, in analyzers, which are flow cytometers that analyze but do not sort cells, the stream goes from bottom to top, and the laser intercept is within the quartz flow cell, whose optics are optimal for sensitivity of detection. In the center, we have a classic stream and air sorter. The stream is from the top through a nozzle, and the laser intercepts the stream as it passes through air, so some of the fluorescence is lost as it passes through air to the detection system. And finally, we have the hybrid sorters, which combine the optical advantages and sensitivity of the quartz flow cell with the stream in air that's necessary for sorting. Laser beam geometry will affect the accuracy of the fluorescence measurements. As you can see here, the highest laser power is in the center of the beam. Beam expanders are often used, which widen the highest power area of the beam. And most instruments now use top hat or flat top lasers, which have a broader and more uniform power distribution. The reason for the importance of a broader and uniform laser beam is that the fluorochromes on the cells must be as uniformly excited by the laser as possible in order to minimize the variation and maximize the accuracy of the emission measurements. We can see here that the beam expander is effective in increasing the area of the high power center of the laser to a certain degree, but it will not give us as wide and uniform excitation as the current flat top lasers. The pressure differential between the sheath pressure and the sample pressure will affect sample resolution, so the amount of pressure applied to the sample tube will affect the quality of the fluorescence measurement. Remember that the sheath pressure stays fixed and is regulated by the cytometer, but you as the user regulate the sample pressure. If you run at a low sample pressure, say here the sample is running at 12 microliters per minute, the sample is pushed into a very narrow core stream within the sheath stream. This means that all of the cells pass through the central highest power area of the laser beam, and their excitation and emission are very uniform. If you push the pressure up, here at 70 microliters per minute, the core stream is wider, and the cells are dispersed across that wider core, so not necessarily in the center of the laser beam. Excitation and emission are less uniform. The result, as we see in a sample of beads here, is that at low sample pressure, the bead peak is very narrow and uniform. This is essential for applications like DNA ploidy and S-phase, where resolution of close peaks is necessary. At high sample pressure, The same beads give a much wider distribution, which will reduce resolution between peaks, but it's perfectly adequate for many applications, such as immunophenotyping. Problems can arise with samples that are clumpy, have a lot of debris, or if air bubbles get into the system. Here you can see a sample running with the cells dispersed normally across the core, and we have a nice tight peak of sample in the expected position on the scale. Here, however, there's a clog from clumps of cells or debris, and it's causing turbulence, diverting the cells to the side of the core. Excitation's reduced because they're not going through the center of the laser beam. Thus, the fluorescence emitted is lower, and it gives an artifactually low and wide peak. This kind of issue can ruin a valuable experiment and can be avoided by good sample preparation and rigorous instrument cleaning and maintenance. Quality control beads should be run every day and whenever a problem is suspected. Now let's take a look at optics. We will discuss two kinds of optics, excitation and emission optics. The excitation optics consist of lasers, lenses, and prisms that focus the laser beam on the spot where the cells will pass through. The lasers used for excitation in flow cytometry have the advantage of being bright, coherent light, emitted at a single wavelength. They're very stable and can be focused on a tiny area like a sample stream. In the last 30 years, lasers have evolved from huge, expensive water-cooled lasers to becoming very small, powerful, much cheaper, and are available in many wavelengths. That said, here again are the lasers that are standardly found on most instruments and which excite the most commonly used fluorochromes. The classic blue 48 laser excites fluorochromes such as FITC, PE, GFP, Alexa 488. The red 633 is good for APC, Alexa 700, APC Cy7. The violet 405 is good for brilliant violet and all the other violet excited dyes. DAPI, which is a DNA-specific dye. Yellow-green lasers at 561 are optimal for PE and its tandems, cherry and red fluorescent proteins. And finally, the UV and near-UV lasers are good for the brilliant ultraviolet dyes and other UV-excited dyes such as Herxt or Indo for calcium flux. And these are only a few of the fluorochromes available that are excited by these lasers. The excitation optics, the lenses and prisms, direct and focus the laser beams so that they converge on the flow cell where the cells pass through. Here we can see a blue laser beam, a yellow-green, a red, and a violet, all converging on the flow cell. The lasers are focused at different points on the stream. The cells will pass through laser 1, then laser 2, laser 3, and laser 4. The collection optics are the lenses, mirrors, and filters that collect the emitted fluorescent light as the cells are passing through the lasers, then separate the different wavelengths and direct them to the appropriate detectors. The emitted fluorescence and the side scatter light are first collected through a lens coupled to the flow cell, as in an analyzer where the flow of liquid is from bottom to top, or at a short distance from the stream, as seen in the stream and air sorter here, where the flow of liquid is from top to bottom. After collection by the lens, the emitted light then passes through optical mirrors and filters, which separate the different wavelengths and direct them to the right detectors. Let's now take a look at the different kinds of filters we use to accomplish this. Here we have a long-pass filter, which lets through only the wavelengths above its designated wavelength. This one is an LP500. LP is for long pass, 500, which means it lets through all of the light above 500 nanometers. Similarly, here we have a short pass filter, which lets through only the wavelengths below its designated wavelength. This one is an SP500, SP is for short pass, 500, which means it lets through all of the light below 500 nanometers. Bandpass filters let through a specific range of wavelengths. They're designated BP for bandpass. The number after BP is the center wavelength of the window it lets through, and the number after the slash designates the width of that window. This means that a bandpass BP 530/30 lets through 530 plus or minus 15, which is half of 30. Thus, it lets through the wavelengths 515 to 545. There are a lot of bandpass filters available targeted to your fluorochromes emission spectra. Most cytometers will have the filters adapted to the standard fluorochromes. Dichroic filters or mirrors are the same long pass and short pass filters, but they are angled so that the wavelengths that should pass through, and the rest are not lost but reflected towards other filters and detectors. It's important to know the excitation and emission spectra of your fluorochromes and to know your instrument to ensure that you have the lasers needed to excite your fluorochromes and the appropriate filters for their emission. If you Google spectral viewers, you'll come up with many options online. Most of the cytometry vendors have a spectral viewer, and they allow you to visualize and download the spectra of the fluorochromes, as well as the laser lines and the filters. Let's look at an example of how these dichroic mirrors and filters are used to separate out and direct the different wavelengths to the light detectors. Here we have a very simple two-color system. The cells are stained with FITC, which emits green, and PE, which emits orange. They pass through the laser, fluoresce, and then the light is collected by a lens at 90 degrees. The first filter they encounter is an SP 560 dichroic, which allows all emission wavelengths that are below 560 through, and directs everything else, that is above 560, to the right, towards the PE detector. The light detectors are colorblind. They only see photons of light, so we must ensure that the photons of light that they see are of the wavelengths of the fluorochrome that we want to measure in that detector. The bandpass 585/42, directly in front of the PE detector here, only allows the wavelengths 585 plus or minus 21. That is from 564 to 606 through to the detector, and this corresponds to the optimal part of the PE emission spectrum. If we look up here, where it's green, the wavelengths lower than 560 are passing through the dichroic towards the detector for FITC, which also has a bandpass BP 530/30 in front of it to refine the wavelengths to those corresponding to the optimal part of the FITC emission spectrum. Here is a slightly more complicated system where we've added in forward and side scatter. You'll remember that the forward scatter is collected in the axis of the laser beam. So here on the right is our forward scatter detector. The bandpass filter in front of it is the same as the laser wavelength, 488 nanometers, as it is scattered laser light, not fluorescence. The side scatter and all the fluorescence are collected by the lens at 90 degrees. They pass through a 510 long pass dichroic, which lets through everything above 510 and reflects everything below 510 to the left, towards the side scatter detector. Again, as the side scatter light is the same wavelength as this 48 laser, there's a bandpass in front of the detector that allows that light through, a BP 48810 that lets through 483 to 493 wavelengths. Up here, everything above 510 then encounters a 560 long pass dichroic, so that everything above 560 goes through, and below, the wavelengths between 510 and 560 then are directed to the green detector with its 530/30 bandpass in front. And finally, the wavelengths above 560 arrive at a mirror and are reflected to the orange detector with its 585/42 bandpass in front. Here's an actual version of the previous slide. We are looking at it from above. Here's the laser, focused on the flow cell. The forward scatter filter and detector are on the right. At 90 degrees, the light passes through the collection lens, then is separated by the 510 long pass towards the side scatter detector with its 488/10 bandpass. Up here, the 560 long pass dichroic sends the green emission that's below 560 to the FITC detector with its 530/30 bandpass. And lastly, the mirror sends 560 and above to the PE detector with its 585/42 bandpass. This is the schema for the caliber optics on the left. On the right is a photo looking from above. Here's the collection lens at 90 degrees to the laser. These are the dichroic mirrors, and on the left we have the light detectors, in this case PMTs or photomultiplier tubes, which we will discuss in a minute. There are many ways of using these mirrors and filters. On the left we have a linear array, sometimes called a boulevard for obvious reason, which uses short pass dichroics to separate and reflect the wavelengths. On the right is BD's octagon system, which uses long pass dichroics to collect the longer wavelengths first, and then bouncing the rest across the octagon, ending with the shortest wavelength detector. And of course, there's always a bandpass to refine the wavelengths in front of each detector. Up until now, we've only been looking at the light emission from a single laser. Let's look at what happens when there's more than one laser. The lasers are separated in space, focused at different points vertically on the stream. The cell is coming down the stream and first goes through the 488 laser. The fluorochromes that are excited by the 488 fluoresce, and the emission is sent to the light detectors. The cell continues, then passing through the red laser, where the fluorochromes excited by the red laser fluoresce and their emission collected. In this case, the distance between the first and second laser is such that the cell passes through the second laser 30 microseconds after it goes through the first laser. Thus, the delay between the two lasers, in this case, is 30 microseconds. Here we have 4 lasers: laser 1, laser 2, laser 3, and laser 4. The cell goes first through laser 1, then the second, third, and fourth lasers at specific times, different for every instrument, and determined by laser alignment, the distance between the lasers focused on the stream, and the sheath pressure, how fast the sheath fluid is moving. The interval between when it passes the first laser and the next laser is called the laser delay. In this case, the cell passes laser 1 at time 0, laser 2 at its laser delay, which is 25 microseconds, then laser 3 at a laser delay of 65 microseconds, and laser 4 at 105 microseconds after the first laser. In most of the multi-laser cytometers, the light emitted by the fluorochromes as they pass through each laser is collected through the lens at 90 degrees, and at the level of each laser emission is carried by fiber optic cables to the appropriate detector groups. In this case, they are boulevards. Here we have a BD system with the lens and the fiber optic cables carrying the emissions to the octagon or trigon dedicated to the fluorescence emission excited by that laser. There are a few instruments where the lasers are not separated vertically in space. They are focused on the same spot in the stream. These are called collinear lasers. The cells go through the lasers at the same point on the stream, and all the fluorochromes are excited at the same time. We'll talk about the repercussions of this in a few minutes. This 3D schema shows the caliber, excitation, and emission optics with the blue and red lasers, the flow cell, and forward scatter detector. Behind is the collection lens, dichroic mirrors, and in front of each of these light detectors, which are photomultiplier tubes, is a bandpass filter. In the last 10 years, there have been newcomers that are taking over the flow cytometry scene called spectral flow cytometers. They differ mainly in their collection optics, which are much more extensive than the conventional flow cytometers we've just been looking at. A conventional flow cytometer, which is what we've been looking at up till now, will measure the light excited by the fluorochrome's main laser, and the emission is collected in one detector through a bandpass filter corresponding to the highest part of its emission peak off of that laser. Here, PE is best excited by a 561 laser and measured through a 585/40 bandpass. It can also be nicely excited by a 488 laser, although as there is less excitation, its emission will be lower. Spectral flow cytometry collects the whole fluorochrome emission spectrum from all of the lasers. Most spectral cytometers have upwards of 50 light detectors. Here we see what's called the fluorochrome spectral signature for PE. the emission of PE is collected through a series of small consecutive band passes from all 5 lasers. Let's look at how this is done. This is the optical system of a 4-laser Aurora spectral cytometer. For each laser, you can see there is a series of detectors that have consecutive narrow bandwidth filters in front of them, so that the complete emission spectra of the fluorochrome is captured in all of these channels. The first laser here is the UV, and that light emission from the UV excitation is measured in 16 detectors or channels, starting from 365 nanometers, which is just above the 355 UV excitation wavelength, all the way up to the far red wavelengths at 829. The bandpass filters are very narrow, with widths that range from 15 to 34 nanometers. Similarly, for the violet laser, all the violet excited emission is collected in 16 detectors, from just above the 405 laser excitation, the emission from 420 up to 829. For the 488 blue laser, 14 detectors collect that emission from 498 to 829, and for the 640 red laser, 8 detectors collect the emission from 652 to 829. This is a 5-laser instrument, and these are the filter and detection arrays for collecting the light emission excited by each laser. The emission wavelengths are, of course, higher than the excitation for that laser, so that for the UV excitation, the emission collected starts at 365, just above the 355 laser wavelength, and goes all the way up to the far red, which takes 16 detectors or channels. Whereas for the red laser, the emission collected starts at 652, just above the 640 laser wavelength, up to the far red, which only takes 8 detectors. The emission spectra from all of the detectors from each laser are collected, and these emission spectra are stitched together to show the intensity of all the emissions from all of the lasers in a single graph. This is called the spectral signature of that fluorochrome. It gives much more information about the spectral qualities and the emission of fluorochromes than conventional flow cytometry. We spoke earlier about choosing fluorochromes whose emission spectra do not significantly overlap. The spectral cytometers, however, do allow us to use many fluorochromes that highly overlap and which would not be distinguishable by conventional flow cytometry. Here we have 2 fluorochromes, APC and Alexa 647. On a conventional cytometer, they're excited by the red 640 laser, and as you can see here, both emit in the red around 660 with highly overlapping emission curves, so they can't be distinguished from each other. But if we look at their full spectral signatures using a spectral cytometer, we can see that while their main emission peaks off the red laser are very similar, the emission from excitation off the other lasers is quite different. The UV, the violet, the blue laser, and the emission off the yellow-green laser is significantly lower for Alexa 647. The significant differences between the emission curves excited off these other lasers allow us to distinguish these 2 fluorochromes. Their full spectra are different, and they can thus be used together on a spectral instrument, where we see here good resolution of co-expressed populations stained with CD8 APC and CD57 Alexa 647. This means that there are more fluorochromes available to be used on a spectral cytometer compared to a conventional cytometer, allowing us to measure more cellular markers simultaneously. That said, there are some highly overlapping dye pairs that cannot be easily distinguished even on a spectral instrument, and you will need to assess these pairs on your instrument and staining panel. A similarity index can be calculated for overlapping fluorochromes that will give an indication of the feasibility of using them together. There are several kinds of collection optics on the spectral instruments. On many of the instruments, the same dichroics and bandpass filters or mirrors we find on the conventional cytometers are used. Here we see the Aurora with its boulevard of mirrors and filters. The Symphony, Bigfoot, and FaxDiscover all use dichroics and bandpasses in different configurations, large or small. A couple of the Sony spectral cytometers use a series of prisms to separate out the wavelengths and direct them towards the light detectors. And the Sony ID7000 uses a light diffraction grating to separate the wavelengths. Now let's look at the electronics. The electronics consist of the light detectors, amplifiers, and converters, which measure the photons of light emission from the different fluorochromes and convert them to a digital number proportional to the amount of fluorescence emitted as the cell went through the laser. The information is then saved to the computer in a data file. Here we have an overview of the electronics, and we'll look at each of these steps in more detail. The fluorescent light emitted by the cell passes through the optical collection system. Here we see the final bandpass filter, so that the photons of light arriving at the detector are of the wavelengths that we want that detector to measure. The light detector emits a current that is amplified, producing a voltage pulse that is proportional to the photons of light that reach the detector. The voltage pulse is digitized to a channel number, again, that corresponds to the amount of emitted fluorescence seen by the detector, and that number is saved into a data file. Here we see that cell number 1 has a measurement of 600 for its FITC fluorescence. Now we'll look at these light detectors. There are several different kinds of light detectors. Simple photodiodes, which are relatively insensitive detectors and are used for strong light signals like forward scatter. Photomultiplier tubes, or PMTs, are highly light sensitive and used for side scatter and fluorescence. On the left is the classic older large PMT that we find on the Caliber and the LSR2 and the older sorters. The newer smaller versions are currently used on many instruments, and on the right is a PMT array consisting of 32 PMTs used in some of the new spectral instruments. Avalanche photodiodes, or APDs, are also highly sensitive, particularly in the red wavelengths, and are now used in many instruments, both spectral and conventional. And last are the newer silicone photomultipliers, which are the next generation of highly sensitive light detectors and already being incorporated into some of the latest instruments. Let's look in depth at how a PMT works. Here's the PMT with its photosensitive cathode. The photons of emitted light hit the cathode, which then generates electrons. The electrons then pass through a dinode chain, which is a series of dinodes with voltage applied, which multiply the electrons at each stage so that many electrons reach the anode for each electron that left the cathode. The anode produces a current, it's amplified, and a voltage pulse is generated that's proportional to the number of photons that hit the cathode and to the voltage that's applied to the dinode chain. You control that multiplication factor by changing the voltage on that detector using the instrument settings in the acquisition software. Depending on the voltage applied, it's possible to have a multiplication factor of as many as 10 to the 7th electrons at the anode for every electron that left the cathode. In the avalanche photodiodes, or APDs, the incident photons generate electron-hole pairs, and a gain is applied that induces more electron pairs to create an electron avalanche and a current which is proportional to the number of incident photons and the applied gain. So on an APD, it is the gain that is applied that increases or decreases the output voltage pulse size and detector sensitivity, similar to the voltage that's applied to a PMT. The APDs are very sensitive, particularly in the red wavelengths. How is a pulse created on a flow cytometer? On the right here, we have a graph of time on the x-axis versus voltage pulse on the y-axis. As the cell enters the laser beam, the fluorochromes excite, and the emission begins to arrive at the light detector, which generates the beginning of the voltage pulse. When it's in the center of the laser beam, the whole cell and its fluorochromes are maximally excited and the voltage pulse reaches its peak. As it passes out of the laser beam, the voltage pulse drops to 0. There are 3 measurements we can make on the voltage pulse. the area of the pulse, which is the measurement from the time the cell enters until the time it exits the beam, the height of the pulse, when the cell is maximally excited in the center of the laser beam, and the width, which is the amount of time it took for the cell to traverse the laser beam. Depending on the instrument, area or height is standardly used to quantify fluorescence emission. The width measurement is generally used in combination with area to distinguish doublets or aggregates from single cells. The analog to digital converters, or ADCs, transform these voltage pulse values into a digital channel number. Here we have a series of large and small voltage pulses. We're looking at the pulse heights, which range from 1 to 10 volts. These voltage height values are converted and binned into digital channel numbers. The smaller pulses will be binned into the lower channel values, and the bigger ones into the higher channel values. The number of bins or channels on the scale depends on the ADC, the sampling frequency and resolution number of bits. Let's look at this again. On this instrument, there are a maximum of 1024 channels. A small pulse is binned into the channel at 300, the medium at 600, and the large pulse is at 800. A reminder that the voltage pulse size and final channel number depend on both how much fluorescence emission reached the light detector and the voltage or gain you applied using the software to the detector's multiplication system, either voltage for a PMT or gain for an APD or photodiode. We'll look at the effect this has in a moment. Remember that the cells go through the lasers one after another, so these light emissions from the cell, as it goes through the series of lasers, reach the detectors and generate pulses that are staggered in time. The cells must pass each laser at the expected laser time delay to be fully quantified. Here the cell goes through the blue laser at time 0, red at 13 microseconds, and violet at 22 microseconds. The electronics are set to measure those pulses at windows of time dependent on the expected laser delay, and then it will correlate the measurements for all of the fluorescent emission from all of the lasers for each cell, and it will save that in the data file. For each cell, the voltage pulses from all of the detectors are digitized to a channel number, and these values are coordinated and stored cell by cell in a standard format data file. The saved data file lists scatter and fluorescence values for each cell, and that can be for millions to tens of millions of cells. Let's discuss the effect of changing the detector or voltage gain settings on your data. Remember that the size of the voltage pulse is proportional to the amount of light hitting the detector and also dependent on the voltage or gain you apply to the detector's multiplication system with the instrument acquisition software. The forward scatter detector can be a photodiode, a PMT, or an APD depending on the instrument. The detector settings will either be a voltage if the light detector is a PMT, or a gain if it's a photodiode or APD. Here we have the same cell being measured, and we're running it at 3 different settings. On the left, the setting is 300. The generated voltage pulse is small, and the channel number it's digitized to is low. The same cell run at 400 has a bigger pulse, digitized to a higher channel number, and if we run it at 500, the pulse is huge and the channel number is very high. So you can see that while the cell is emitting the same amount of light each time, and the same amount is reaching the detector, how you choose the voltage or gain applied to that detector will change its multiplication factor, and thus the size of the pulse, and where the cell will fall on your scale. Threshold is another crucial element to consider in setting up your instrument and your experiment. All cytometers have a threshold to determine what's considered an event, cell, bead, particle, and what is not, what you want to be considered as background or debris. The threshold is the level above which detected signals will be processed and saved. There's always a threshold, even if you haven't specifically set one. While threshold can be set on any detector, it's usually set on the forward scatter parameter. So if a pulse is lower than threshold, it will not be seen or processed. Anything below threshold is excluded from acquisition display, from the saved data file, and any downstream analysis. Let's talk a little more about threshold. Here, as is most commonly done, the threshold is set on the forward scatter. We have the same cell here run at 3 detector gain or voltage settings as on our previous slide. But now we have shown the threshold as a pink line. The plots below are forward scatter on the x-axis versus side scatter on the y-axis. Again, the small pulse at 300 is digitized to a low channel number. The pink vertical line is where the threshold is set. We're showing the dot of the cell at 300, but in actuality, because it's below threshold, it will not be seen or counted. The 400 and 500 pulses digitized to higher channel numbers are above the threshold and thus will be visible and saved in the data file. This is a real-life example of the effect of changing the threshold. This is a peripheral blood sample, so a mixed bag of cells and possible debris all run at the same voltage or gain setting. You can see that there's a wide range of pulse sizes corresponding to the range of cell sizes. Here again, the threshold is set on the forward scatter. On the left, the threshold is set very low, difficult to see, but just below the first mark on the forward scatter x-axis. The smaller pulses are above threshold, and you can see that population on the lower end of the x-axis. These could be red blood cells, platelets, or just small debris. Above them, the lymphocytes are visible, then the monocyte population, and finally the granulocytes at the top of the scale. In the center, the threshold has been increased, and a lot of the small pulses are below threshold. On the forward versus side scatter plot, you can see where the threshold line cuts that population in half. Everything below threshold is lost, both visually and in the data file that will be saved. You can tell by that sharp cutoff that you're losing events below the threshold. On the right, the threshold is higher still, and that whole population is now gone, and no indication it might be there. The cells of interest for the researcher are most likely the lymphocytes or larger cells, and the exclusion of the little stuff may be justified here. But if it was platelets or red blood cells that they were interested in, then they're lost. Setting up the instrument for detector voltages or gains and threshold is crucial to assure that you're including and acquiring data on the cells you want without too much debris. It's important to be very careful here, because if you set up your trigger threshold channel, usually forward scatter, with the voltage or gain too low, and the threshold is set too high, you may not see and you may lose your cells of interest. Another issue to be concerned about if you're sorting cells is that since the instrument does not see what's below threshold, it cannot exclude them from the population you're sorting. Debris or unwanted cells that are below threshold will wind up in your sorted populations, which can be problematic depending on your downstream application. Data files are acquired and stored by the software in the cytometer's computer. Each cytometer has its own acquisition and analysis software. For instance, BD has Diva, Cytoflex has the Site Expert, the Aurora has Spectral Flow. There are many third-party softwares available for post-acquisition analysis, such as FlowJo, FCS Express, Kaluza, and these are more commonly used for data analysis than the cytometer acquisition software. The FCS data file format is standardized across the industry so that they're readable by pretty much all of the data analysis software packages. The saved data file lists scatter and fluorescence values for each cell and event, contains instrument settings and cytometer information, as well as experiment and sample identification and annotations. It's important to annotate all the pertinent sample and experimental information into the data file when you are running your samples and acquiring the data. It's saved with a file, and then it can be retrieved or searched when doing post-acquisition analysis, maybe a year or two from now when you've totally forgotten what you did. The data can be displayed as single-parameter frequency histograms with statistics available on how many cells in each population, how positive or negative they are, and as dual-parameter plots, any parameter versus any other parameter, which allows deeper identification of different subpopulations. There are lots of options for how to display one or two-dimensional data, as shown here. The standard dot or pseudocolor plots, but also contour, density, and zebra plots sometimes help to distinguish small populations. Most instruments acquire and save the data as linear. The total number of channels on the scale is dependent, as we discussed earlier, on the ADC, the analog to digital converter. The data, however, can be displayed on graphs as linear or log, or as we will discuss in a minute, by exponential scale. Linear scale is standardly used for forward and side scatter, and for specific applications such as DNA ploidy, where the range of intensity is relatively small, and it is important to visualize small differences in the fluorescence intensity of populations with different DNA content. On the right, we're looking at log scale, which is needed when the dynamic range of fluorescence intensity of the different populations is very large. Log scale allows the visualization of both the very dim and the very bright populations on the same graph. If the display had been linear here, one or the other of these populations would be off scale. And on the bottom histogram, you can see that there are very many low events piled up in the bottom channel. Of note is that in the older analog instruments like the caliber, linear or log scale amplification is done in the hardware. Once the data is collected as either linear or log, it can't be displayed any other way. Log scale can be problematic due to pileups in the bottom channel, as we've just seen. Here we see again, in both the histogram and the pseudocolor plot, that there are many events on the bottom axis. There are a number of reasons for these below zero values for a cell. On many instruments, there's a background subtraction function that diminishes dim positive values. There are corrections applied due to spectral spillover, and as we've seen earlier, it's crucial to have the correct detector voltage or gain settings so that all the cells are on scale. It's now recommended that all data, except for specific linear applications like we just discussed, be displayed on a biexponential scale, sometimes called hyperlog or logical. We can see here the scaling around and below zero. And the cells that were piled on the bottom of the log axis are now visible in biexponential. Biexponential scaling transforms the data so that the values are shown as log, down to around zero, where they're shown as linear, and farther down return to log. This allows everything below zero to be visualized, and there are often whole populations down there that would be missed otherwise. This visualization of log versus biexponential scale can have a drastic effect on the identification and the statistics of the populations of interest and can falsify your experimental results. Here we've drawn a gate on the log display to cover what we see as the CD8 positive, CD4 negative population with a percent positive of 19.7. If we apply that gate to the biexponential display, the percent positive is the same, but we can easily see that the gate is not encompassing the whole population. If we enlarge the gate to encompass the whole population, our percent positive is more correctly 23%. And the same holds true for the CD8 negative, CD4 positive population. One of the major advantages of flow cytometry is the ability to use sequential levels of gating, which allows you to look at multiple small subpopulations and to look at changes in these populations comparing control samples to experimental conditions or normal samples versus disease. Now let's talk about spectral spillover. We have seen that the cytometer optics are carefully designed so that each detector only sees and measures a certain range of wavelengths that correspond to the excitation and emission maximums of the fluorochromes that we want to measure in that detector. All very nice and tidy, but in reality the fluorochromes don't play fair. As we have seen, the fluorochromes have a range of excitation wavelengths and a range of emission wavelengths that can be wide and do not necessarily restrict themselves to the range of a bandpass filter in front of one detector. Let's take a look at some of these issues as they affect our measurements in a conventional cytometer. Let's look first at spectral spillover due to adjacent emission spectra. Here are the emission spectra of FITC and PE. You can see that FITC's main emission peak is at around 520, and is captured by the detector with the 530/30 bandpass, which is the primary detector for FITC. You can also see that its emission spectrum overlaps with the PE emission spectrum, so that FITC has significant emission out in the orange wavelengths in the range we use to measure the PE fluorescence with a 585/42 bandpass in front of the PE primary detector. Most of the FITC emission will be measured at 530, and it will be brightest in its primary detector here, emitting a large voltage pulse. But some of the FITC emission, that is in the orange range around 580, will go into the PE detector and will elicit a smaller pulse or signal. This means that in the detector that we want to measure only PE, we are also measuring some FITC. Here we have a single stain control, cells that are stained only with FITC. There's no PE added. You can see that there's a nice positive and negative on the FITC x-axis, but also on the y-axis in the PE channel. So it looks like this is positive for both FITC and PE, but there's no PE in this tube. So all of that fluorescent spillover is coming from FITC, and we'll talk about how we handle this in a minute. Another significant source of spillover is cross-laser excitation. Here we have two fluorochromes that emit in the same green range, cyan and FITC. Their emission on most conventional instruments is collected through a 530/30 filter. They differ, however, in their excitation spectra. Cyan maximally excited by the violet 405 laser and FITC by the blue 488 laser. They are also excited by the other lasers to a lesser extent, particularly FITC by the 405, cyan a little bit by the 488. In this conventional cytometer with spatially separated lasers, we see a 488 and a 405 laser, and the detectors off each laser are measuring the fluorescence through a 530/30 bandpass. The FITC stained cell passes first through the 488 laser, its optimal excitation, and the FITC fluorescence elicits a signal in the detector off the 488 laser that's dedicated to FITC. It then passes the 405 laser, where it has less but significant excitation, and the FITC emission elicits a smaller signal in this detector, which is dedicated to cyan. So we have spillover from the FITC into the cyan detector due to cross-laser excitation. In a conventional cytometer with collinear lasers, the lasers are not spatially separated, but are focused at the same point on the stream. Here we have a cell stained with both FITC and cyan, which emit at the same wavelengths. They are passing through and being excited by the 488 and 405 lasers at the same time. The detector has a 530/30 bandpass and only sees photons of those wavelengths, but cannot tell which fluorochrome they originate from, so we don't know if the signal is from FITC or cyan. So the signals from fluorochromes that are excited by different lasers but emit at the same wavelengths cannot be separated and thus cannot be used together. This limits the number of fluorochromes that can be measured simultaneously on collinear laser conventional instruments. To recap, there are three sources of spectral spillover: adjacent spectra, where the emission spectra from fluorochromes excited off the same laser partially overlap; tandem to base. Remember we talked about tandem dyes, two fluorochromes coupled together. If the coupling is not optimal, there can be incomplete energy transfer and some leakage of fluorescence from the base or donor fluorochrome. Here it's spilling into the PE detector. And as we just saw, cross-laser excitation between pairs of fluorochromes that have similar emission spectra but are excited off different lasers. Here, BV711, mainly detected off the violet 405 laser, but is also to a lesser extent excited off the 355 laser, so it will spill into the detector for BUV737, which has similar emission but is optimally excited by the 355. Indeed, spectral cytometry takes advantage of this cross-laser excitation to distinguish similar fluorochromes and makes use of all the emitted light across all of the lasers. Here is one of the bad boys, PE Sci5 Tandem. Nice and bright in its own detector, it spills over into multiple other detectors by all the means possible. Adjacent spectra, cross-laser excitation, and tandem to base. This is only one fluorochrome. The more fluorochromes, the more spillover. We need to accurately measure only one fluorochrome in each detector, so we have to assess and remove the spillover coming from other fluorochromes. And here is the spectral signature for PE-Sci5 on a 5-laser instrument. Maximum emission is off the yellow-green laser around 670, but you can see that it is excited by all of the lasers and emits into most detectors. So what do we do to get a true measurement of each fluorochrome's emission? First, we use single color controls. These are cells or beads stained with only one fluorochrome at a time. The contribution of light emission from each fluorochrome into all of the detectors must be measured using single color controls for every color in the mix. Then we apply mathematical corrections. Mathematical algorithms based on the emission of these single color controls into all of the detectors are calculated and then applied to the multicolor experimental samples to enable accurate measurement of each fluorochrome. This applies to both conventional cytometers and spectral cytometers, although the methods are slightly different. Both rely on single color controls. In a conventional cytometer, each fluorochrome's emission is based on one dedicated detector, its primary detector. The single stain controls establish the amount of spillover into the other detectors, and the software uses matrix algebra and inverted spillover matrix to calculate the corrections and subtract the light contribution from other fluorochromes so that the primary detector is accurately measuring only the dedicated fluorochrome. In a spectral cytometer, each fluorochrome is measured in all of the detectors. The single color controls establish reference spectra for each fluorochrome, and a linear unmixing or deconvolution algorithm establishes an unmixing matrix to determine which combination of reference controls best fit the signature emission of the multicolor samples. While we're not going into detail in this course on compensation and unmixing, here's a quick overview of how it's done. Let's first look at compensation in the conventional flow cytometers. Here's our single color control that has only FITC added. On the x-axis for the FITC detector, we have a nice FITC negative and FITC positive population as we should. But if we look at the y-axis for the PE detector, we have what looks like a PE positive population. Looks like it's a double positive, with a median channel value of 5,000 on the PE scale. But there is no PE in this tube, so we know that this is due entirely to spillover from FITC at these wavelengths. This allows us to calculate and apply a compensation correction to this data, so that in the PE channel, the median of the FITC positive and negative populations are the same. The FITC positive population is now PE negative, as it should be. Note that it's correctly compensated, but there's generally a wider spread on the corrected FITC positive population than on the negative. All of the acquisition and post-acquisition softwares now have compensation algorithms that will do this automatically. On the multicolor sample, here uncompensated, it looks like there are several double positive populations. Once the single colors have been run and the compensation applied, it appears that this population is single positive, it's negative for CD8, and this one is negative for CD3. And there are two real double positive populations. Spectral cytometers use linear unmixing or deconvolution. The single color controls are run and the signatures saved. Here is the multicolor, in which the cells are stained with both FITC and PE. And here you can see the curves for the double stain sample with both FITC and PE, the FITC and the PE single colors. The unmixing algorithm uses the single color signatures to determine what proportion of each fluorochrome best fits the emission curve of the multicolor samples. We've looked at what flow cytometry is, what we can measure, and how a flow cytometer works. Let's review what we've learned. Light scatter. Cells or particles pass through the laser and scatter the laser light in all directions. We measure the scattered light in two directions, forward scatter in the same axis as the laser beam, and side scatter at 90 degrees to the laser beam. The forward scatter gives an indication of the size and the surface properties, while the side scatter is more dependent on the structural complexity and granularity. This allows us to distinguish different populations of cells or particles based on refractive index, size, surface properties, internal complexity, but cannot be used to quantitate exact size. Fluorescence and fluorochromes. Fluorochromes are molecules which can absorb light energy at one wavelength and then re-emit that energy at a longer wavelength. Each fluorochrome has its own excitation spectrum, across which it can absorb light, and its own emission spectrum, across which it can emit light. The emission wavelengths are always higher and have lower energy than the excitation wavelengths. Many fluorochromes are now available. The newer ones are brighter with narrower emission spectra. Fluorochromes are used coupled to antibodies, bound to cellular organelles, or intrinsic to the cell, and the amount of fluorescent light emitted by the cell or particle as it passes through the laser gives a measure of how much fluorochrome and thus how much of the marker of interest is on or in that cell. Another look at our simple fax caliber with fluidics, optics, and electronics. The fluidics system. Sheath fluid and sample are pushed through the flow cytometer by different means of positive or negative pressure. Hydrodynamic focusing ensures that the cells pass through the center of the laser beam to be optimally excited by the laser. The sample pressure is controlled by the user. Higher sample pressure can increase the variation in measurements. Clogs and air bubbles can deflect the flow of cells to the side of the laser beam and cause artifacts in the data. The importance of good sample preparation and instrument cleaning cannot be overemphasized. Quality control beads should be run every day and whenever there seems to be a problem to ensure that the cytometer is functioning correctly. The excitation optics consist of lasers, lenses, and prisms that focus the laser beams on the spot where the cells will pass through. The collection optics are the lenses, the filters, and the mirrors that collect the emitted fluorescent light, separate the different wavelengths, and direct them to the appropriate detectors. Here you can see the laser focused on the flow cell, the collection lens at 90 degrees, and the mirrors and filters directing the light to the detectors. The fluorescent light emitted by the cell has passed through the optical collection system, and the photons of light arrive at the detector. We've looked at different kinds of light detectors, from simple photodiodes to PMTs, APDs, and silicone photomultipliers. The light detector emits a current that is amplified, produces a voltage pulse proportional to the light that reached the detector and to the voltage applied to the detector multiplication system. The voltage pulse is digitized to a channel number, and that number is saved into a data file, cell by cell, with a value for every detector, and the data can then be analyzed for identification and statistics on subpopulations of interest. The spectral cytometers have many more detectors than the conventional cytometers, allowing them to collect all of the light emitted by each fluorochrome across the whole emission spectrum for every laser. The emitted fluorescence is separated into consecutive narrow bandwidths of wavelengths, either by the optical filters and mirrors similar to conventional cytometers, or using prisms or diffraction gratings, and directed to the light detectors. The light emission measured by all of the detectors are collected and stitched together to show the fluorescent signature from all of the lasers in a single graph. Fluorochromes with similar excitation and emission spectra, which are not distinguishable and cannot be used together on a conventional cytometer, can be distinguished and used together on a spectral instrument. Another advantage of spectral flow cytometers is the ability to measure and subtract the autofluorescence of different types of cells, which increases the sensitivity when the specific staining is weak. We have seen that there are three sources of spectral spillover that can result in fluorochrome emission into multiple detectors. Single color controls allow the evaluation of fluorescence emission into all the detectors and the calculation and application of mathematical algorithms to ensure the accurate measurement of the emission from each fluorochrome in a multicolor experiment, either by compensation in a conventional instrument or by linear unmixing or deconvolution in a spectral instrument. The advent of the new spectral instruments currently allows upwards of 50 markers to be measured simultaneously, and those numbers are expanding every year. And of course, this makes for massive data files and more information than the human brain can easily understand. The new high-dimensional data analysis tools being developed facilitate interpretation of these complex multicolor datasets. A big thank you to my friends and colleagues who've generously donated their best slides over the years that have been so helpful in putting together this talk. Bill Telford, Paul Wallace, Al Salek, Grace Chanoski, Rai Fuchel, Paul Hutchison, Kylie Price, Maria Hymas, Krishna, Celine Lage, and Sherry Thornton. And a huge thank you to Bill Telford, Rai Fuchel, Paul Wallace, and Jeff Osborne for their valuable time and expertise in reviewing this talk. And again, my thanks to the ISAC Education Committee reviewers, Kathy Daniels-Klupfel, Jello de la Cruz, and Evan Jellison for their helpful comments and suggestions. And may the flow be with you.